Again. It's an honor to be here today with two people who have led the effort at Massachusetts General Hospital regarding xenotransplantation. I am here today with Tatsuo Kawai, who is the director of the Legorreta Center for Clinical Transplant Tolerance, and he is professor of surgery at Harvard Medical School. He is the A. Benedict Kazemi Endowed Chair in Transplant Surgery. And also with Leo Riella, who is the medical director of kidney transplantation, and he is a physician scientist. Both are close colleagues and friends. I'm Camille Cotton, and I'm the clinical director of transplant infectious disease at Massachusetts General Hospital. So it certainly has been an exciting week with the announcement of what happened. So could you tell me a little bit more about what motivated our team to pursue the transplantation of a genetically edited pig kidney?
Actually, let me start first. We have been working with eugenics in preclinical studies using non-human primates. And after transplanting their pig kidney to the non-human primates, then we found we can achieve the over 2 years of survival. So that moment, I think we decided to, go to the humans. So the experience in the preclinical study is one of the reasons. And the other reason is that I went down to the National Kidney Foundation, for the patient to hear the patient voice. Then at that meeting, I realized that patient really wants us to go ahead. So with some encouraging results in preclinical studies, plus patient's strong desire to go ahead, that push us to go ahead.
That's great to hear, and great to hear how patient-focused it was, even though it is some pretty heavy-duty science. Can you tell us a little more about the genomic editing that was done? Why was that necessary? And then how does it move the field of xenotransplantation forward?
So obviously, triple knockout, right? So three major carbohydrate xenoantigens removed. That's been done, but eugenics pig has 7 human transgenes, two complement related genes, and two thrombosis coagulation related genes, and 3 of the inflammation or immune related transgenes. We don't know, actually, no one knows which human transgenes very essential. But what we have done is triple knockout without any human transgenes or 7 human transgenes. It's obviously different. It's without human transgenes, all animals most lost kidney function by 1 week. So it's very consistent results. But the other group with human transgenes survived longer, like over 1 year.
Yeah, I would just add that the gene editing revolutionized xenotransplantation because it allowed to do all these genetic mutations in multiple genes with precision in a short period of time. And if you look at the survival of xenotransplants before gene editing and after, it was in the days to weeks, and then suddenly it was months to years. So in a short period of time and a few years when those techniques were applied, it just completely kind of changed the field. And I'll just add one more thing. Tatsuo had, Dr. Kawai had the best preclinical data out there in kidney transplantation. And we know that xenokidney does much better than heart and lung and liver. So kidney is the easiest one, even though it's still challenging. But as we started to see, number one, how the public was reacting to the xenotransplant being placed on a brain dead donor and not having that reaction that many of us, were worried about that there was going to be, a negative impact about using those genetically modified pigs. And suddenly, with the impulse of patients just, knocking our doors and asking, when are we going to be able to get a kidney transplant from a modified genetic pig, and bypass this waiting list that can be devastating to many patients. So I think there was, all these different, fronts that were just, colliding and making, like the real moment, I think, for us to try to make that first step. And it's a small first step, but I think it's a first step in the right direction.
Great, great. Well, certainly there's been a lot of effort at MGH over the decades looking at transplant tolerance. Can you tell us a little about what you needed to do for induction immunosuppression, and then what your plans are for maintenance immunosuppression in this specific case?
So my impression while working on preclinical studies, it's still hypothesis, but I have an impression that for xenotransplantation, the medication against the innate response, to suppress innate response, is, of course, is essential. However, once we overcome innate response, adaptive response is relatively weaker than those against the allotransplant, alloantigens. That's what I'm feeling, because we were able to stop tacrolimus completely after 6 weeks in monkeys, after which only anti-CD154 and Cercept is a maintenance immunosuppression. So it possibly is totally different from the allotransplant. It can be less maintenance medication is necessary for the xenotransplant. We don't know. We have to carefully observe those patients. Then we have to establish the immunosuppressive medications protocols.
Yeah. I may add that, in addition to the gene editing, the use of the anti-CD154, so this co-stimulation blockade that back in the days, when I started in the lab in the 2000s, it was supposed to be the blockbuster, the drug that's going to change how we manage our patients. But unfortunately, the first drugs that were tested in humans led to this thrombosis. And it took a few years for them to modify the molecule. And now it's getting back to the clinic. And this drug was really the one that, on the preclinical models that Dr. Kawai and others have used, have changed that survival, had increased even further. So the genetic editing plus this co-stimulation blocker with the CD154 that attacks both innate and adaptive immunity seems to be kind of the secret sauce to be able to get those long-term survivals. And then for the rest of the maintenance, as Dr. Kawai mentioned, the tacrolimus, myfortic, and maybe a low-dose prednisone doesn't have to be that high. And this is where, we are currently, aiming for our patient to have a quadruple treatment, but lower dose, and potentially even consider weaning some of these agents long-term.
Yeah, in addition, the reason why CD154 is important, we also tested the CD40, anti-CD40, but anti-CD40, the results were much worse than, much inferior to the CD154. So because CD154 can also block CD11B, which is very important for the inflammatory response. But CD154 can block both the CD40 and CD40 ligand pathway and the CD11B. So that's why CD154 is very important for xenotransplant.
Great. It sounds like you will learn so much from this. What plans do you have moving forward, and how will you incorporate the learning of xenotransplantation into some of the other work you're doing with tolerance and other areas?
Well, in terms of tolerance, the ultimate goal of xenotransplant is also tolerance, but it's very difficult. I think my mentor, David Sachs, is always, telling me we should do a tolerance for xenotransplant. But so far, adaptive response can memorize, right? Since they memorize, we can induce tolerance because memorize against the transplant organs to be tolerant, right? But innate is usually cannot memorize. So some recent studies saying just like even macrophage can memorize, but basically neutrophil cannot memorize, right? So um, if they don't memorize, if they don't have a memory function, it's very difficult to induce tolerance. That's not my so. But eventually, if we can do all more, genetic modification, put the more human genes, then we may be able to, if we don't need to worry about any innate response, then only to have to take care of adaptive response, maybe tolerance induction much easier than the allo transplant.
And I think it's a great debate in terms of, how many mutations do you do? Can you add more things to, regulate the immune system? And when do you decide that that is enough? So I think it has to find a balance, right? So I think you have to target the things that we know are completely incompatible. So we knew the complement, the coagulation. There were two things that if we didn't target, those kidneys had a really high risk of having, issues with clotting. So once you solved all those, what else is important? I think infectious disease, safety of these kidneys. So they inactivated 59, porcine endogenous retrovirus, and for many, including Dr. Fishman, they believe that this was important. This added, an extra level of safety when using these types of kidneys. So I think we now have to learn with what we have, because, based on the preclinical evidence, I think it's really hard to get better than a couple of years in a primate. Even a couple of years, it's outstanding. Remember, they don't have all the diagnostics that we do in terms of managing these patients. They don't have all the drugs that we use in terms of managing complications. So I think just being capable of achieve that was a green light for thinking about the next step into a human trial.
Yes, excellent, excellent. So, Leo, how do you foresee this amazing breakthrough impacting the future of organ transplantation and addressing the existing organ shortage?
Yeah, I believe xenotransplantation will revolutionize how we take care of patients with advanced organ failure. I think in the kidney world, because priority on the list is determined by waiting time. So if you get on the bottom of the list, independent if you're doing poorly or okay on dialysis, you're going to wait the same amount of time to get to the top, slight variations according to blood group. But this is, on one side, it's quite unfair. And because some patients that are doing very poorly, they probably won't make to the top of the list. So I think being capable of having an alternative and deciding, okay, which patients, the risk benefit may indicate that we should give them a xeno versus keeping them on dialysis, because most likely they won't make it until the top of the list. And even if the xeno becomes a bridge, even if it's not going to be much better than the human kidney, which, if we're optimistic, we hope that it will be, potentially slightly worse. But still, seeing it as a bridge, I think it would be incredible.
Absolutely. Patients always talk about time off dialysis as being a real gift. So even if it's not perfect, it probably would be better than more dialysis, we would hope. And so we're all active members of the Transplantation Society and think a lot about the global transplant community. Do you think this is something that could be scaled up on an international level eventually? Right now, it's extremely, research intensive, and obviously you've gone through a lot of regulatory approval, local regulatory approval with the IRB and the Food and Drug Administration in the United States. Do you think that eventually this is something that would be scalable, including to middle and lower income countries?
I think so. Only thing you just have to make sure is eGenesis has one advantage, right? They inactivated the PERB. Although PERB has been said is no concern, but that was an additional safety uh, guarantee. So that's why we are confident about using that modification. So if we until we know everything about infection, maybe we should not spread too much. We just have to watch carefully, and if I'm sure it's fine, but just have to be prudent in terms of infection. Then we can go. So, but I have to count on you how to monitor infection.
Yeah.
So.
But I think it can have a huge impact because of the finances of dialysis, right? Dialysis it has this extreme cost. So if there are multiple companies, as we're seeing now, that are entering the xenotransplant business, and the potential are endless for them to even have different price ranges, developing countries and developed countries and so forth. So, and imagine there would be an initial investment, which is, doing the transplant, but, the follow-up, the drugs, the cost is significantly lower than keeping the patients on dialysis. And not even that, the complications that dialysis cause in the long term, and the hospitalizations and so forth. If you put on the pen and write it down, I can clearly see a path forward. Once we, of course, have enough safety data to move forward, that this should be spread beyond, developed countries.
That's really wonderful to hear. And it's always a good perspective to think about the cost of dialysis, which is quite significant. And in many middle and lower income countries, transplant is advantageous because it's more cost effective. So it's interesting to think about scaling this up in those regions. So let's move on to closing remarks. What do each of you want the international transplant community to know about your work? Any closing thoughts or concerns or things you'd like to share, each of you? I'll call on Tatsuo first.
Well, obviously, we closely monitor our patient, and the results will be officially reported to worldwide transplant societies, and that is our obligation, right? Then, but Leo, can you add something?
Yeah, no, I think, our hope is that this will bring, some excitement in attracting new talent to transplantation, right? So I think as transplant nephrologists, I have to say, I think one of the worst part of our jobs is going through a selection meeting where we have to choose patients to place on a list. And at the same time, we're removing patients on a weekly basis because they either die or because they're too sick now to get a transplant. Not only that, they have to come to our office and we spend 30, 45 minutes to tell them why we feel that transplant is not an option for them anymore, while 2-3 years before we said it was. And so I really feel that is going to, inspire, I think, many of us to try to push this forward for our patients. We gotta reach out to them and say, look, this is something that we need you guys to advocate for. As physicians, we can advocate up to a certain level. The impact of a patient advocating for something like xenotransplantation has a completely different impact in the regulatory agencies, in the government, in NIH, and so forth. So we need to get our patients out there to be advocates so that we can move this forward, and safely, of course. But I think there's huge potential, and I think this is going to benefit all of us, and most important, it's going to benefit our patients.
Great. That's a wonderful perspective and a great message.
One thing to add is that we can do human patients slowly, but enrolling human patients slowly. But we, at the same time, we still have to do preclinical studies a lot. I saw one patient visited to my clinic today. She's 100% sensitized, and waiting for identical, 100% matched donor for 23 years, but never got transplants. So, but we still don't know those 100% sensitive patients can get the xenotransplant. So those HLA antibody can cross-react to the graft. We still don't know. So next thing we really have to do just preclinical studies. Those kind of things needs to be clarified in preclinical studies. Then we can do it. Apply to the xenotransplantation to the sensitized patient. So it's still long-term process.
Sure. Well, that sounds like a great opportunity, especially for those sensitized patients or people who might not otherwise get organs. What do you anticipate over the next few years as far as the ability to scale this up? What type of time frame will you move in as far as enrolling new recipients?
I don't know. Leo may be thinking differently.
No, it's challenging.
I think
it doesn't depend only on us, right? It depends on the companies. It depends on the FDA. And so I think if everything goes well, continues to go well, we hope that we're able to convince, both the companies and the FDA that we need more of these patients getting transplanted so we can learn as much as we can to advance this. So we want to do it carefully. So of course we want to watch it for a few weeks, months, and confirm and try to find the best regimen so we can scale it. But I think we got to start the dialogue yesterday because there's a need.
Great. Well, thanks so much to both of you for speaking with me, and I look forward to hearing your presentations at the Transplantation Society meeting in Istanbul this coming September. By then, you should have a lot of really interesting information to share with us. And wishing you both good luck, and thanks so much for all your hard work getting us to this time.
Thank you.
Thank you, Camille. Bye, everyone.
Bye.